I welcome everybody. On behalf of the Southern California section of the American Chemical Society, we call it SCALACS. We are very honored to have Dr. Arieh Warshel today make the presentation. And I am also thankful to Professor Eric Stemp, Arno Papazian of Santa Monica College, Professor Eric Stemp of Mount St. Mary's University, and Nancy Brown of California Academy of Math and Science for agreeing to host this function, representing the local colleges and the local high schools and the school district. And I also thank the local section actions committee of the National ACS for awarding us a DEIR, that is the diversity, equality, integrity and respect. That is the DIR. So I thank the local section action committee of the National ACS to give us a grant under this DIR program to conduct these functions. And without any further delay, I pass it on to Professor Eric Stemp of Mount St. Mary's to do the introduction, and then of course he will pass it on to Arno. Thank you.
Okay, good afternoon, everyone. I am the co-chair of the Department of Physical Sciences and Mathematics at Mount St. Mary's University, along with my colleague, Dr. Denise Chismajian, who also happens to be Arno Papazian's wife. So we have a little, circle going here. And anyway, we're very honored to be, part of the hosting process for this, lovely event to honor Dr. Warshel. And I'd like to say a few words about Mount St. Mary's because I think we fit in very well with the diversity theme for the grant that funded this. And this is Mount St. Mary's at the Shalon campus, which is in Brentwood. And you can see over here on the left, we're on top of a little mountain. Our former school president, Jackie Dowd, used to say that only God and Mount St. Mary's look down upon the Getty Center, which is a little ways further over to the south and east. And then this is a picture from one of our main buildings out towards the center of the campus. And it's a lovely place where we work. The Mount has been kind of at the forefront of diversity for quite a long time. Before diversity was really a buzzword, the Sisters of St. Joseph took it upon themselves in the late 60s, early 70s to kind of revamp the mission of the school, in their words, to work towards the education of the immigrant daughters of Los Angeles. And that became a major part of our mission. And just along with that, I just wanted to share a couple little photos. In the sciences here, we're very proud that even though we are a small school, that we take research very seriously, and especially in our department, we have a lot of active faculty in research, a lot of students. And this is a picture of several of our students from about 10 years ago. This is me about 10 or 12 years and 10 or 12 pounds ago. And all of these students have now graduated. A number of them have gotten medical degrees, pharmacy degrees, and are working in chemical industry and so on. This was a very hardworking group of kids, and as you can see, it's obviously it's a very diverse group, and we do take our students to meetings every year. We try to go to the ACS meeting, and this, for example, was our foray to the ACS meeting in 2009 in Salt Lake, and it's some of the same students. So we work hard and we play hard when we go to the ACS meeting as well. But on that, I think I'll just kind of say, we're proud to be a part of this, and thank you very much. And Dr. Papazian, would you like to take over?
All right. I don't have any screen to share. I'll just talk. I have the privilege of introducing Professor Arieh Warshel today. And I spent 4.5 years with him as a postdoctoral researcher. And this was the period that had the most impact on my maturation as a scientist. We spent countless hours, countless lunch and coffee breaks together talking about science, politics of science, as well as other aspects of life. And I learned a great deal during those conversations. And I hope I was a useful sounding board. Now, the description in the flyer says that he's considered an out-of-the-box thinker. He's certainly that, but that does not entirely capture the context in which he worked. Professor Warshel spent a large chunk of his scientific career trying to show his colleagues where the box should be. His record of publications not only presents significant advances in our true understanding of biochemistry and biophysics, but also they chronicle a scientist's struggle to convey the truth to others whose careers wouldn't necessarily easily benefit from a change in the prevailing approaches. During my time with him, I saw that scientists are not immune to groupthink or snap judgments. Working against the grain, Professor Warshel had to convince his colleagues, one by one, patiently, over decades, of the value of his ideas, his approach, and his work. He won the 2013 Nobel Prize in Chemistry, along with Martin Karplus and Michael Levitt, quote, for the development of multiscale models for complex chemical systems, unquote. Translated, a significant motivator for awarding the prize was incorporating quantum mechanics into classical simulations in order to study chemical reactions. His success in incorporating the most essential aspects of quantum mechanics into classical simulations and cleverly capturing the electrostatic effect of the environment on the reaction allowed him to truly understand how enzymes actually catalyze reactions that would otherwise be extremely slow. This was at a time when such studies were regarded either as impractical or impossible, and the prevailing thoughts about the basis of enzyme catalysis were largely missing the point. Daring to dream big and working on that dream by bringing to the table a bright mind along with a fierce perseverance in the face of constant prejudice are some of the great attributes that contributed to Professor Warshel's success. Now it's time for us to hear his story from Professor Warshel himself.
Okay, I would like to first thank the two introductions, especially Arno. I remember that one we tried to respond to a negative referee report. Sorry, we wrote the report on what solvent mean, and we really needed the Oxford Dictionary for it. But we had a lot of discussion about science and the futility of fighting too hard. And I enjoy one paper with Arno where we were invited for an ambush in discussion about electrostatic, where the idea was that everybody will write a paper and then send it and there will be no change. And then we realized that this was an ambush and we succeed somehow to provide a new version, which is very deep and also respond to all the different minds. But let's go to this story. So most of it is summarized in the very recent book that I wrote, From Kibbutz Fishponds to the Nobel Prize. And I will go through some key points from this book and my kind of life story. Now let's try to open it. Okay, so we start with my parents. My mother came from a city that probably only Anna knows where it is, though yesterday I saw it on the news. It is now in Ukraine. At that time, it was in Poland. And my father, on the other hand, was from another city in Poland, which is now in Belarus. And both of them have relatively different background. My mother was from a better family. She went to gymnasium and to started a university. My father did not finish, I think, elementary school, but he was self-taught and extremely smart. And both of them decided in the 30s to immigrate to Israel. They were in Zionist movement, and they found kibbutz called Sde Nahum in 1937. My mother worked at the beehives and then different other occupations. My father worked for a long time in the fish ponds, which were a major part of the kibbutz economy, and later he became an accountant in the kibbutz. I was born, so this is another picture. I was born in 1940 and grew up in the kibbutz in what is called children house, because they have this idea that the kids should be together and only limited amount of parents' time. I enjoyed this life form very much, but once in a while there are books and people who complain that this was very traumatic. A lot of the story on childhood in the kibbutz were made up in this book of Bruno Bethlehem, who was professor at Harvard, and he spent maybe 2 weeks in Israel and wrote this book, suggesting that kibbutz children have no ambition because they go all together, which of course was opposite from the reality, but it was a highly quoted book. So I grew up in the kibbutz, and I done several scientific experiments. I parachuted cats, but Peter, people don't get excited. It was only from 2-story building, the only high building in the kibbutz. And they would have made it anyhow without the parachute. I also try to design airplanes from balsa, which were supposed to be propelled by flies. This experiment did not work too well, but you try and sometimes you fail. What is this? Okay, so I grew up in the kibbutz with a combined class of basically 2 classes. And one part which was not so great in the kibbutz was that they did not emphasize school. So it really depends on your inclination and ambition. They had examinations, but there was very little to enforce the examination, which means that people who did not study, the only punishment was not to go to the movie, which was shown once a week outside. At the last year, fortunately, we had a joint class from 4 other kibbutzim in kibbutz called En Harod. Well, this was one of the nicest years in my life. We studied much more, but still none of these studies, or most of it, was not aimed at the matriculation, which was essential for being accepted to the university. And we study a lot this type. I mean, we study theoretical books, but it was the theory of socialism and communism from different directions, including the short course of Stalin, but also different theories, a lot of literature, and a little less physics, though last year, the oldest teacher in Israel, which was from Kibbutz Harod, is still teaching, he is 90-something, so he mentioned that I study in his class. I almost don't remember it, but he attribute part of his teaching to my Nobel Prize, and this is fine. So after the kibbutz, we all went to the army. I attended officer course, and I stay one more year in the standing army because I had to somehow pass external matriculation, since they did not accept what was taught in the kibbutz as a matriculation or anything related, and the kibbutz had no major interest to teach you, so you will leave the kibbutz, though there was the idea that you could go to university, but for each year in the university, you will owe 2 years in the kibbutz. So I regrettably left the kibbutz, and then I went, sorry, this is another picture from the army, and I finally succeed to pass the matriculation, not such a great grades, but I pass it. And then I succeed to pass the concourse for the Technion, Israel Institute of Technology, and was accepted there. And basically, I knew that I want to go to university, but I had zero idea what direction to choose. It's not like kids now that, you know, keep asking what will happen if I go there. I basically believe that eventually you will find your direction. And I choose chemistry because my friend from the army told me that I have good eyesight, and therefore I could see color and I should go to chemistry. So this was the information that I received, and I accepted. I start chemistry, just trying to pass all the courses to get high grades, but somehow toward the third year, I start to be interested in enzyme. This is enzyme that, as you see, and I will show you again, it does chemical reaction much faster than the same reaction in water. And this is used, so you will have to do chemistry in the time that is needed for the body and not to wait forever. And it so happened that I choose a kind of project with a professor named Yechiel Shlitin, and he was working on chymotrypsin, and I decided to measure this relatively fast reaction. And for this, I had to wait until the only NMR at the Technion had an averaging device, and I succeed to make maybe the first measurement of fast reaction in enzyme, but I did not publish it because I felt that it's not good enough. But one notable thing that he insisted that we will submit some abstract to the Israel Chemical Society, and in this abstract we concluded that electrostatic is not important in enzyme catalysis, which was of course also accepted by many other people, and it's obviously, I spent a large part of my life to show that this is wrong. So I also spent some time in the summer in the, how you call it, agricultural engineering or soil engineering, and I had to do measurement of diffusion, I did huge amount of calculation by hand, and then I start to learn that there are computers in the Technion with a paper tape, but it was too late, so I did all the calculation by hand, but I registered in my mind that it could be great to use computer one day. And now this is one of the random decisions that I made, which kind of guided my life. When I was looking where to move, I read Israeli newspaper in the weekend edition, and they told about a new scientific director of the Weizmann Institute named Schneur Lifson. And what attracted me in the story was that originally he was among the founders of a kibbutz, which is about 2.5 kilometers from my kibbutz. Their kibbutz had the distinction to be the first in the what was called a tower and stockade, Choma Migdal, which was a way that this settlement were built in one day, so they will be able to defend themselves. My kibbutz was about 2 weeks later. So, and they also wrote that he come to work on a bicycle. So I decided to go to see him. I went with my, I think, girlfriend or almost wife, Arieh Warshel. We went to his office. I show him my grades, which were almost straight A. And he, of course, say, I have no interest in grades and I don't take students. And nevertheless, he took me to his group, which had only one other person. And in 1966, I joined his group. And I start to work on trying to use computers to model molecules. He just moved from analytical work on helix coil transition to believing that computers would be useful. And I took the project of trying to design what will be called force field or molecular mechanics. I start to work on this, and the program that I start from it did not work at all. I mean, because it was based on internal coordinates. And with internal coordinates, it's very hard to do first and clearly second derivatives analytically, because each bond length could depend in a ring on many other torsional angles. So I kind of gave up, and then I discovered that if I use Cartesian representation, suddenly everything becomes trivial. And I was able to figure it out because at that time Israel had a computer, it was just built, called the Golem. Maybe I'll jump to it. Sorry. Yes, there was a computer called the Golem. Golem is this legendary robot that was built in Prague to help the rabbi to do the Shabbat work. And this is the name that Professor Pekaris, who built the machine, chose for the computer. And it had the distinction of having 18 significant figures. So I could do perfect first and second derivatives, and I realized that I could check anything. I mean, now it looks trivial by using numerical derivatives. And suddenly I could do normal modes of any molecules without spending 1 year reading the book about normal modes, which I spent a year reading. And I start to realize that with computers, things that look impossible, because people at the time always try to find simplifications for some derivatives, like what would be in electrostatic, the famous Gauss theory or Maxwell equations, I found out that if you just start from the first equation and try to formulate it in the computer in the simplest way, you don't have to know all these theoretical things, because all what they were aimed is to save a little time, and now you have a machines that help you a lot. So we develop a force field. This is representing atom as ball and springs. And at that time, suddenly the 6-day war erupted. So I was called to the army and spent some time on the Golan Heights. And when I came back, we joined by Michael Levitt, who came before his PhD. He was told by Kendrew that he had to join our group. And we developed a general program based on the Cartesian idea, which allowed to treat basically any molecule and to calculate first and second derivative, vibrations, crystal structure, and so on. And it was called at the end the consistent force field. So this is again me near the Golem in 2013. At that time it was a museum piece. And more or less the time of my PhD stopped, but with this force field we could do a lot. The program became eventually the way to model proteins with very few changes. But one thing we could not do, and this is to describe chemistry, breaking bonds, because if you have ball and springs, you cannot describe the process that when you break a bond, electrons move to one atom and leave the other one. So starting with some games in 1969, it evolved, and eventually in 1976, we have a very influential paper that show how to combine quantum mechanics and molecular mechanics in treating enzymatic reactions, because I was, since the Technion, very interested in enzymatic reactions. And again, it was far ahead of its time. This work showed how to treat electrostatic effects in proteins, how to represent the induced dipoles, and a very simple representation of the solvent, but it was nevertheless explicit representation. So this was a part of what is called multiscale modeling, where the idea, which follow me all the time, was that you represent the part that you can in high level. It could be quantum mechanics, but you never forget about the rest of the system. If you want to study airplane, it's not enough to have fantastic description of the chair. You need a description of the whole airplane. This might look obvious today, but I have a long story on referee reports, which says that my model of water, which was up to infinity, is not a complete Hamiltonian. I never understood this statement, where it was the most complete Hamiltonian one could think about, but it was simplified at the longer range. And some significant work was done when I spent 2 years at the Medical Research Council with Mike. This is my 2 daughters and my wife in Cambridge. And at that time, we completed several works. One of them, a simplified model for protein folding, where we replaced the side chains by spheres. And it was used later in many incarnations. We also published the QMMM at that time. In another work, which was very pioneering, was using surface hopping, which at that time was used for reactions of at most 3 atoms to describe photochemistry in large molecules. Later, we used the same approach to describe electron transfer in solution. In this approach, you run trajectories on a whole system, classical system. It could be a solvent or large molecule, and treat the transfer between 2 electronic states quantum mechanically. So it's a 2-state quantum mechanics, but all of this evaluated along the trajectories of the whole system. And fortunately, this was the first molecular dynamic simulation in biology. We got 100 femtosecond at the time when nobody knew what is femtosecond. Essentially, it was known that the visual process occurred in less than 6 picosecond. So we got basically exactly the time which was later observed experimentally. And we also discovered huge surface crossing, which was basically finding conical intersection, again, at a time where I have no clue what is conical intersection. By the way, I also have no clue what is coarse grain, which was the name for a lot of these approaches. But so it's paid to know names, but it's my not strongest point. So we modeled the first step in the visual process, the isomerization in rhodopsin. And it was done. After we done it, we done a movie. At the time, still at the time when in Hollywood people use this plastic with many, many pictures. It was done with an Hollywood person. And what you see here is a trajectory on the excited state, crossing to the trans direction and relaxing. We will see it one more time. So the trajectories were taken from the 1976 papers, but the production was done in 1980. And very few people believe it. Essentially, I gave a lecture in a Gordon conference of crystallographers. And when I finished, one of the audience asked me, what evidence you have for this? I was so shocked that I didn't know what to answer to him, but let's go further. Eventually, this story was in part in an Israeli stamp that was given after the Nobel, exactly at the time of the Year of the Light. So if you have this envelope, you could read the story on the back of it. Another nice discovery, because we knew how to model enzymes, but there were a lot of items that we have to figure out. And in a meeting in the Vatican, you see here John Paul and my wife, I published a paper in the Pontifical Academy on how you do free energy perturbation in enzymes. So at least one holy publication. Okay, so now back to more detailed items that we figure out during the year. Back to now modeling enzymes, now with all atom simulations, because time passed, and with free energy perturbation. So one of the systems I like to show is the enzyme RAS gap, which is involved in hydrolysis of GTP to GDP, which is part of a signal transduction cycle. And when it has wrong mutations, you get cancer. So there are several chemical steps. The whole issue of phosphatidrolysis had thousands and thousands of experimental papers with huge number of professors, and all of them believe in experiments, which are fine, but it's very hard to do experiment on the transition state. And during the year, I become more and more convinced that, I mean, there is this idea that George Olah used to quote, that you could only disprove a theory, but you cannot prove them. But with computations, you could eventually go to the details of mechanism and figure out what is right. It's clearly happening in small gas phase reactions. So the way we do it now, we run the reaction first in solution with QMMM simulation. This is hydrolysis of GTP with the help of a magnesium. You get ideas on the free energy surface, and then you transform the surface to the protein by an approach that is called the empirical valence bond approach, which basically fit different diabatic state to the overall ground state found by the ab initio calculations and transfer it to the enzyme. An anecdotal story on this, that when I studied in the Technion in the 4th year, I joined physicists, though I was chemist, and they keep talking on the Born approximation, Max Born, which always end up with some asymptotic wave function. I could not completely follow this Merzbacher book story, but I told the physicist that one day I will have asymptotic wave function for enzymes. 15 years later, I think that the EVB fulfilled this promise. So, of course, you could get nice movies from it, of the dynamics of the reaction. This is the reaction of breaking the GTP in the enzyme, where water molecule transfer a proton to the phosphate with the help of the green magnesium. And what is much harder is to get from it the free energy surface. This is really a major challenge because you have to do a lot of sampling. And we done it for this and many other enzymes. We also address the question of what really is the origin of enzyme catalysis. Usually it's 1 hour lectures. There are probably 30 proposals. Most of them are between incorrect to completely incorrect. And we spend a lot of time trying to show which approaches are incorrect. And we were able to do it because of the computer, because in almost no case you could really even define the question without being able to dissect the contributions. And we end up, these are many enzymes that we study where you see the activation barrier in water and the activation barrier in the proteins. And in all cases, almost all cases, it was electrostatic in nature. I would say all cases. And basically, the enzyme always provide complementary charge distribution to the transition state. And it does it by trying to minimize the reorganization of the environment. This is somewhat complicated story, so I wrote maybe 50 papers on it, and still most of the community don't get it. Essentially, it's very joyful to write recent papers that attack this concept without knowing what it is, because it basically say that the catalysis is not due to the interaction of the enzyme dipoles with the reacting fragments, because similar interaction exists in water, but with the fact that in the enzyme you don't have to change the orientation of the dipoles too much during the reaction. But we don't have time to talk about it. We use this approach to model mutations of different residues in enzymes. This become more and more important during the years. This is example in dehalogenase, where we try to reproduce the effect of different mutations. And I will comment about it a little later. Now, another direction that become more and more interesting is what to do with large systems that work in very long times. And it goes like this, that when you have small system, you could run explicit Newton laws on the atom, molecular dynamics, get the free energy. You could also now use very powerful computers like this Anton of Shaw et al. But still there is a lot to do with smart approach. In one direction we did is exploiting this simplified folding model where you represent the atoms, the side chains by spheres. We add to this approach the electrostatic aspects. So we were able to study more functional aspects. And during the year, we also found a way to model long-time processes where we have to use Langevin dynamics or Brownian dynamics instead of molecular dynamics to be able to describe very long-time processes. And I will show you very few examples. We have countless of them. One of the first was the action of ATPs. It's a molecular motor, a very smart motor, where the chemical energy of ATP hydrolysis lead to rotation of a central stack and allow it to do work. And one of the issues that we start first to attack, and we decided that we must understand it on the same level that first or second year physics students understand how such a motor works, how, let's say, the water flow allow us to pull up a weight. So here you have a very complicated system, but we want to understand it by the rule of physics rather than by poetry and nice words. I mean, a lot of biophysics and biochemistry are described like songs, so you don't really sure what people are talking about, but you clearly cannot quantify it. So one of the things in this motor was that scientists were able to measure the rotation of it by putting probes and seeing how fast it rotates, and found that it does not rotate all the time in 120 degrees, but it stops every 80 degrees, and then it continues to 120. So by having this simplified model, free energy, we were able to see that the free energy landscape is such that first you could rotate to 80 degrees, then you do the chemistry, and then you move to 120. So this was something that you absolutely cannot, or I don't see how you could explain experimentally. We also generated landscapes that explain how the overall energetics of this motor works. It takes like 5 minutes to explain it, so I won't do it now, but remember that if you want to understand how such molecular motor work, you must have a landscape. Otherwise, what you say has no meaning. And in later years, we tried on different ATPs and found that they have different landscape. But in all of them, the end result is that you have the same torque. So we got more and more understanding. We understood, for example, that it's not the 10 kilocalorie per mole that you release during the chemistry that give it directionality, but the fact that the surface is tilted in the right direction. And again, I don't have time to talk about it, but we believe we describe the system on the level of this story. And one more example, this is kinesin type system from the myosin family that apply in your muscles. There is a question why it goes in one direction. Now, there is a huge amount of experimental study of such systems, but none of them ask why it moves in one direction and not backward. Because if you draw the different steps, you could find out that the same way you go forward, you could also go backward. So you must find what happened. And a lot of the discussion in this field are based on metaphysics when people say that there is power stroke, so it sounds to you really very impressive, that kind of throw the foot forward, and this is why it goes in one direction. So first of all, you could have the same story going backward. Second, it defies microscopic reversibility and many other stories, yet most of the scientific community believe in the power stroke story. So we spent some time trying to debunk it, and here, eventually we found out that what makes it go in one direction is that the ADP release is slower in one direction than in the other, and that the protein is really open more if you try to go backward. So if you try to go forward, then backward. And we did simulations that reproduce this unidirectionality, again, with a simplified coarse grain model. And this story is still not finished, but again, it's very hard to explain people that when they make argument, to tell them that, look, unless you talk in our language, which sounds very arrogant, until you talk in the language of physics, we don't know what you are talking about. Okay, we've done a lot of studies in explaining ion channels. You see, when the polarization is as described here, the ion cannot pass, but when you change the polarization, the channel open and the ion pass through. Okay, we've done a lot of movies of this. Maybe we show you for 1 second, just nice movie. How okay, I forgot to put the movie in. So this is another example, but now a few words on another directions that we now push a lot. We wrote countless papers on enzyme and why different mechanisms work and do not work, but it seems to have an impact, but not sufficient considering how right we are. So you have to do miracles, like to make prediction, and then people believe you. Like people start to read Newton Principia only after Halley predicted that the Halley comet will come in the right time. So we try to do enzyme design and to predict the effect of mutations. We had limited success, better than others, but not good enough. And we found that this is absolutely a major challenge, because if we know different mutant on the pathway of, let's say, directed evolution, we could get quite nicely the activation barrier. But when we try to move from one mutant to another by simulations, we have limited success. One of our latest example was to generate many mutants, creating this strange funnel, and we show that the best mutants in the pathway of directed evolution are among those that we predicted. Very recently, show you later, another direction that we made a lot of progress was GPCR, which are the ways that the cell get the signal from the outside. So usually it works. This is adrenaline receptor. The adrenaline binds from the outside. This activates GDP, replaced by GTP, and the signals start to be transferred. So there has been huge experimental progress in this field. And what we done, we use our simplified models to generate the free energies of different intermediates. And this lead to this type of landscape that allow one to try to design drugs and to understand how it works. I'm finishing in 2 more minutes. Recently, we also jump into the COVID front. In about May last year, we start our first study, and we predicted residues that, when mutated, could change the binding of the virus to the receptor. This one, our predictions, essentially appear to predict several of the new variants, like the British mutant, the South African, and so on. In a later paper, we actually show that the mutants that we predicted and some new mutants, how they increase the binding to the receptor and decrease the binding to the antibodies. We also been lucky to predict residues that appear in the Omicron. And we also work on trying to design or to help in designing inhibitors to the COVID. So we design approaches for covalent inhibitors, and we continue to work with this. And the final thing that we've done very recently is trying to use artificial intelligence in maximum entropy to try to find trend in enzyme catalysis, and this is something which is going on very actively. So the early works which were done at the end of the 60s and some other effort eventually in 2013 led to the Nobel Prize. Of course, I have to thank to many collaborators. Some of them could find themselves there, and without them it would be very hard to try to convince people. We did not convince, but we at least try. Forgot what they say. They say something about that you convince people only after you die or they die, but I forgot. It's usually the story. And of course, I want to thank my wife, who is sitting on the left side with the Swedish prince in the Nobel ceremony. So I will be happy to have some questions, so we see what is the next step. Thank you very much. I prefer to, if you ask me the questions out on the chat, I will respond to them. Thank you for the question. Absolutely no, I did not even know in the kibbutz exactly what is science. And also at that time, again, I did not know. I knew at some stage that I would like to enroll in a university, but no clue about science or scientist until the technical. The takeaway is that it does not matter what is your background. If you study, and the emphasis is to study the actual basic STEM professions like mathematics, physics, chemistry. If you have these tools, you could always try to deal with science or with other technological directions. And also not to get discouraged, this is also to college people, not to get discouraged from negative opinions to scientists in general, not to get discouraged from negative referee reports. Essentially, I have very few papers that were not rejected originally. So to have the tenacity to keep your opinions and also to try to do new things which your curiosity guide you to do because they might be important in the future. By trying to, by hatred. No, really, if you get negative referee report, the first 2 days are very hard, and trying to respond to it is a motivation. By trying to show that you are right, this is a very strong motivation. And I think that I have in myself stubbornness, so this kept me motivated. I did not feel stuck because I had always several projects, so always one of them would go. And also by experience, I knew that eventually there will be a solution. In this respect, essentially, the computer helped a lot because you could use it to guide you to
It's
see,
17 hours.
Oh, you could use it to guide you for what works. So I always have the computer to support me.
And we have a lot of great questions in the chat, but unfortunately we're short for time. Dr. Prakash, do you want to go ahead and conclude?
Okay, yeah. On behalf of the Southern California section of the ACS, I would like to thank my distinguished colleague, Professor Arieh Warshel, for this beautiful lecture, talking about his early childhood in kibbutz in Israel and how he developed his whole, molecular mechanics approach to solve important chemical biological problems, the enzyme catalysis. And he has done a fantastic job in giving this talk and made it quite lucid. And I'm very thankful that he gave this lecture to not only us, but also a lot of high school students who participated. I think we had more than 100 participants in this lecture today. And we also had our chemistry department, USC, was also well represented. Thank you very much, Warshel. And thank you all for participating in this important event organized by the SCALACS and also the Senior Chemistry Committee. Thank you.
Thank you.